Bienvenidos al primer seminario.
Ahí está el
acá. Al primer seminario de este año, de esta primera parte del año. Empezamos con un seminario extraordinario, fuera de cronograma, por la visita de los chicos que ahora vamos a presentar. Y van a dar el seminario de hoy. Vamos a tener 3 oradores divididos en 2 partes. La primera parte va a presentar su trabajo Gastón Iturburu. Gastón trabaja en Mar del Plata, en el Instituto de Investigaciones Marinas y Costeras, dependiente de la Universidad de Mar del Plata y también de CONICET. Él va a estar presentando su trabajo que ha venido haciendo en Valle de Uco, en arroyos y ríos, sobre presencia de agrotóxicos y diferentes residuos de origen urbano, y cómo esto puede afectar a la salubridad de esos ambientes acuáticos. Y en la segunda parte del seminario van a presentar el trabajo que han estado haciendo y están haciendo en este momento también en Valle de Uco, a ver si me sale bien, Alex Feckler y Eric Boninger, del Instituto de Ciencias Ambientales, RPTU, de Alemania. No voy a intentar pronunciar las ciudades. Y en este caso ellos están hablando sobre la descomposición por diferentes microorganismos, bacterias, plancton, también macroinvertebrados acuáticos, cuantificando esta descomposición y cómo esto afecta a los niveles tróficos superiores, a la productividad primaria y diferentes fenómenos de la cadena trófica o la red trófica. Bueno, Gastón.
Bueno, gracias por la presentación. Voy a hablar en español, sabes que están en inglés. Una aclaración inicial: mi lugar de trabajo, soy investigador asistente de CONICET, pero mi lugar de trabajo es en Mar del Plata. Pero hace ya 4 o 5 años que venimos interactuando con la que es mi codirectora Jorgelina Altamirano de Yanila y con Erika, que vamos al campo y trabajamos y nos metemos con estos proyectos. Estos son unos muestreos anteriores que ya lo tenemos publicado, y ahora, digamos, la presencia de los chicos acá y, digamos, la posibilidad de dar este seminario está relacionada con un proyecto bilateral con IIMyC, que bueno, esperemos que en el futuro podamos mostrar los resultados de eso que van en marcha. Bueno, lo que vengo a presentar, el título dice plaguicidas y PPCPs. Ahora vamos a aclarar qué son los PPCPs. Básicamente son farmacéuticos y productos de cuidado personal en ecosistemas acuáticos de los Andes de la región central andina, su presencia y una evaluación de riesgo ecológico en el Valle de Uco. Como verán, hay un montón de instituciones involucradas, algunas nacionales, incluso de Córdoba, otras de Mar del Plata, Mendoza y algunas de Valencia, de España, Valencia. Bueno, veamos. No me... Bueno, para tener una idea general de la situación que estamos, ya hace bastantes años que se empezaron a evaluar lo que se llaman digamos, si estamos operando en un espacio seguro a partir de algunas variables, como humanidad y, digamos, el efecto que tenemos sobre el planeta. Este grupo lo venía evaluando hace bastantes años, y si podemos ver este esquema, lo que dice es, si estamos en la parte verde de esa variable, por ejemplo, cambio climático, o la carga de aerosoles atmosféricos y demás, estaríamos trabajando, digamos, viviendo en condiciones seguras para que sean sostenibles en el tiempo para el planeta. Hubo una actualización en noviembre de 2023 y se evaluó también lo que son lo que se dicen novel entities, que básicamente podemos pensarlos como contaminantes o productos antropogénicos que volcamos al ambiente. Y eso abarca un montón de contaminantes químicos, pero también microplásticos y diversos contaminantes. Por otro lado, el Programa de las Naciones Unidas para el Ambiente habla de que estamos en una triple crisis planetaria por el muy conocido cambio climático, también la pérdida de biodiversidad y la degradación de tierras. Esa es la situación en la que estamos hoy en día. Bien, si pensamos, me voy a centrar un poco más en lo que son los contaminantes. Si pensamos que todas las actividades humanas tienen un impacto, y este impacto puede ser a través, por ejemplo, de volcado de productos químicos o uso de productos químicos, podemos darnos cuenta que todo puede terminar en el agua. digamos, es como un pequeño ejemplo. Si pensamos esto como una cuenca que tiene actividades agrícolas, tiene actividades urbanas, vemos que, por ejemplo, los plaguicidas de uso actual, por escorrentía, pueden llegar a los cuerpos de agua, lo mismo los volcados de cloacales. Entonces ahí tenemos esto que yo comentaba al principio, de productos de cuidado personal y farmacéuticos, microplásticos también, o incluso en las reservas de agua, digamos, en algunos diques y demás, se puede dar la eutrofización y se pueden producir las cianotoxinas que son perjudiciales tanto para la fauna que habita ahí como para los seres humanos que usan ese agua como recurso. Entonces el objetivo en particular fue estudiar la presencia de algunos de estos compuestos en el Valle de Uco, donde no había registros hasta ahora de estos compuestos en diferentes cuerpos de agua, y hacer una evaluación de riesgo ecológico. Básicamente tomamos 5 puntos, acá no voy a contarles yo a ustedes Valle de Uco y demás. Básicamente lo que hicimos es tomar muestras de 5 puntos en 2 lugares, en 2 momentos del año diferentes, en algunos arroyos y en el río Tunuyán. Básicamente nos centramos en lo que es el Tunuyán superior, la cuenca superior. Entonces, como decía, se mostró en primavera y en verano. Se hizo un análisis de uso de suelo y cobertura de suelo. Eso no lo voy a comentar ahora por una cuestión de tiempo. Se tomaron muestras de agua, se vieron nutrientes, se hicieron extractos para determinar concentraciones plaguicidas, de PPSPs, se tomaron parámetros físico-químicos, también se tomaron muestras de macroinvertebrados y de la comunidad fitoplanctónica. La idea sería integrar todo eso. El proyecto busca integrar no solo la contaminación química, sino los efectos que tiene en la biota del lugar. Bien. Solo para mostrar algunos resultados de básicamente qué se encontró desde el punto de vista químico. Voy a hacer un pequeño paréntesis para contar cómo es un poco la dinámica que hemos trabajado. Tomamos las muestras de agua, las filtramos acá en el laboratorio de química ambiental, las mandamos a Córdoba a extraer. Eso después las mandamos a España, al laboratorio de la doctora Picó, y ahí se hizo un análisis por HPLC masa a masa. Y luego eso tuvimos resultados, y a partir de esos resultados lo que voy a mostrar también después es una evaluación teórica, al menos, del riesgo que correrían digamos, el ecosistema acuático. En una segunda instancia lo que vamos a hacer es ver si en esas comunidades se vieron afectadas, pero digamos, de hecho, vamos a estudiar la composición de esas comunidades. Está en marcha eso. Bueno, yendo a algunos resultados que quería mostrar hoy, en el gráfico de arriba podemos ver los diferentes puntos que mostramos, o 5 puntos, en primavera y en verano. Y acá lo que se hizo, cada barra es la acumulación de productos, en este caso son plaguicidas, los separamos por mecanismo de acción, herbicidas, fungicidas e insecticidas, y algunos metabolitos, que es el producto de descomposición de ellos. Lo que se puede ver es que no hay un patrón muy claro bueno, en primavera siempre hay menos o más que en verano, o hay un punto que siempre tiene más. Hay unas tendencias, después lo corroboramos un poco con el uso del suelo, donde tenemos franjas de vegetación autóctona alrededor, cubre un poco más, y no es lo que voy a presentar, pero por ahí que le quiero contar esa tendencia que hay. Y respecto a qué compuestos se usan, o qué compuestos se ven, si miramos la parte de arriba, esta barra, por ejemplo, más celeste es de los... imposible. Las barras más celestes son de los fungicidas, y vemos que hay fungicidas como el tebuconazol y el carbendazim, que ya se han informado en muchas cuencas de la región pampeana, la parte más de Santa Fe, en Córdoba, y condice también con alguna información que teníamos de que se utilizan en diferentes cultivos. No solo en los viñedos, sino en ajo, algunos frutales y demás. El metolaclor es un herbicida que se ha encontrado bastante acá. El imiacloprid también es interesante porque, haciendo una revisión de todas de todos los informes que hay de plaguicidas en el país, el imiacloprid aparece siempre. Tal vez se hizo famoso hace un par de años porque tiene problemas con las abejas, con los polinizadores. En Europa está prohibido mucho de sus usos. Y bueno, el carbendazim es otro fungicida bastante encontrado. Ahora bien, ¿cómo transformamos estas concentraciones? ¿Qué quiere decir que haya estas concentraciones? Hay una manera bastante simple, que son los cocientes de riesgo, que es una comparación entre qué hay en el ambiente y cuánto es tóxico para un organismo. Eso es lo que está aquí abajo, ¿sí? Entonces, esta concentración, ¿qué significa en...? Sí, no lo veo. Esta concentración, por ejemplo, ¿qué significa en riesgo? Si tenemos un coeficiente de riesgo mayor a 1, implica un riesgo ambiental o para las especies que habitan el ecosistema acuático. Y como podemos ver, hay bastantes que están cerca del 1, superándolo, y hay un caso muy particular, que es el arroyo claro, a veces lo conoce arroyo grande, es el que pasa por Tunuyán, por la ruta, básicamente, ese fue el punto de muestreo, que las concentraciones de carbendazim aportan mucho al riesgo. Entonces podría ser principalmente se debe a su toxicidad en invertebrados. Ese es un primer paso. Después hay otros pasos para refinar este riesgo, pero como primer pantallazo nos llamó la atención eso. Si pasamos a lo que son los fármacos y productos de cuidado personal, es interesante ver que se han encontrado muchos antiinflamatorios no esteroideos que se usan de venta libre, ibuprofeno, naproxeno, la cafeína, que puede ser no solamente de los fármacos, digamos, sino también de nuestros consumos de café, mate y demás. Tengamos en cuenta que estos productos ingresan a nuestro cuerpo y no siempre son transformados en el cuerpo, y a veces se eliminan de la misma manera. Y las plantas de tratamiento no necesariamente están preparadas para transformarlos. Entonces, como vemos, las diferentes concentraciones, tampoco vemos un patrón. Sí es interesante que los primeros reportes en Argentina de parabenos, que son productos que están en muchas cremas, en la mayor parte de las cremas, y productos de belleza. Y si pasamos a lo que es concentraciones a coeficientes de riesgo, vemos que los coeficientes de riesgo son altísimos. Incluso sin poder tener en cuenta el aporte de algunos de estos compuestos porque tenemos datos de toxicidad. En una segunda etapa, como decía, se va a refinar esto. De hecho, se hizo en verdad, pero no lo estoy presentando. Y bueno, podríamos tener una mejor idea de esto. Básicamente, ¿en qué seguimos? Hay cosas que ya tenemos hechas que no estoy mostrando, que es la segunda capa de evaluación de riesgo, que es por distribución de sensibilidad de especies. Para esto necesitamos muchos datos de toxicidad de especies y no los hay. O a veces tenemos que extrapolar de especies del hemisferio norte. Bueno, análisis del uso del suelo, que puede explicar un poco algunas variables, y un análisis multivariado considerando también variables físico-químicas, ambientales, que pueden estar relacionadas a la presencia de estos compuestos y al riesgo. Esto está hecho. Ahora les voy a meter el chivo de mostrarles el paper, por si a uno le interesa. Y lo que estamos analizando ahora, es, bueno, efectos en las comunidades acuáticas, y eso dentro de este muestreo. ¿Sí? Con los chicos, tal vez algo que van a contar ahora, que estamos viendo si la contaminación afecta a algunas funciones ecológicas, pero hasta ahí sigo yo, después ellos van a hablar de eso. Y bueno, continuar trabajando en red, que la verdad que en los últimos 4 o 5 años nos ha dado bastantes resultados y hemos hecho un grupo de trabajo bastante amplio y de diferentes instituciones con ideas todavía para seguir. Esto seguimos. Y sí, esto representa mucho a veces. Bueno, agradecer los proyectos que han financiado esto, la UIP por financiación una estadía en España, y los grupos de Mar del Plata, de Valencia, el Laboratorio de Química Ambiental de Yanigla y el grupo de entomología, que las veces que he venido me han recibido muy bien. Creo que eso es todo, ese es el trabajo, y cualquier duda que tengan después lo charlamos un rato, si no, me escriben. Gracias.
Me parece que iba a decir, en caso de que haya preguntas, las dejamos para el final, ¿sí?
That's it. Okay, so we are Alex and Eric from the working group of Iturburu, which has the beautiful name Functional Aquatic Ecotoxicology. And yeah, it's working. So our institute is located in the southwest of Germany. And this is the campus. When you want to enter, you have to go these stairs. And we are located right next to the beautiful platinate forest. And one could say that Landau is probably the Mendoza of Germany, because we are the best wine region. The only difference is that we drink the wine in proper volumes, these glasses, and that's now the cultural exchange we have to make. These are called glass. They have like half a liter and these dots that look like a grape. And there's a rule: if you want to drink the wine in there, you have to make 4 fingers of wine and 4 fingers of water. So now you know that. And the idea that we had is that we don't show you one study in depth, but rather show you an overview of what we do and just give you a glimpse into the study. So we constructed this Venn diagram, and really the core or the heart of the working group is, if you see it here in this intersection between ecosystem functions we look at and ecotoxicology. My personal field of specialization is stable isotope ecology, because I'm supervising the lab in Landau for this. And yeah, the first study I want to show is exactly at the heart of the working group. So we do a lot with leaf litter decomposition, because in the headwaters, especially in the Palatinate forest, you have a lot of trees and leaf cover and so on, and it's the most important energy resource. But this process is often affected by chemical stressors, for example, like fungicides. And my personal addition to this research field was looking into the hyporheic zone, so the sediments. And why is that important? First of all, the sediments, they serve as a filter for the microbes, for example, the spores of aquatic fungi, but also the physicochemical parameters change drastically, and we have a completely different community in the sediment, and therefore also a different sensitivity. And the second point is that actually most of the organic matter in many systems is stored in the sediment and not above it. So in the study we found that in the hyporheic zones and the sediment you have a lower decomposition rate, and the importance of fungi gets reduced. And more importantly, we've seen that the fungicide effects were higher in the sediment, which shows that we have currently research bias and we should focus more on hyporheic processes. The second study also focuses on hyporheic processes, but the difference here is that we now look into mainly archaea and the process of methane production. So methane causes roughly 20% of the global climate change, and lots of it is produced from natural systems, for example, the waters. And we asked if actually antibiotics could increase the methane production of a sediment. So we did an incubation study, a small glass system that is anaerobic, and we measured the produced methane and CO2 and found that production rates almost doubled. And we used also stable isotopes for this. So what you can here see is the compound-specific stable isotope ratio, so of the heavy carbon 13 to the light carbon 12C. And an easy interpretation would be that if you're in the top left of this plot, you have more methane produced by hydrogen, and here you have more methane produced by acetate. And we did this over the whole course of the experiment, and you can see that actually the same substrates were used, but the dynamics changed, and therefore we contribute this change in methane production to a change in the microbial community. A study that is rather untypical for a working group is the next one, because here we don't have any chemical stressors involved. It's a purely ecological study where we looked at the food web, the riparian food web, so the flow of aquatic emergent insects and how terrestrial riparian predators like the spiders feed on them. And we had a specific focus on the hunting mode of the spiders. So for example, the blue ones are, see it, these are web building spiders, these are ground hunters, and these are vegetation hunters. And we found that the web builders had the highest aquatic proportion in the diet, followed by the ground hunters and the vegetation hunters. But it was all, for every species assessed, it was all ecologically significant. And a quite comparable study we did with the same methodological approach is the next one, where we looked at a mosquito control agent called BTI. It's applied worldwide, but especially also in Germany, and as far as I'm concerned, it's the only biocide that's actually applied in nature conservation areas. And the reason for that is that it has a very specific mode of action. It should only target Nematoptera, but also the chironomids are affected by it. And chironomids are a really important food source in the aquatic terrestrial subsidy. So what we've seen is that we had different emergence dynamics in the chironomids, which altered the trophic status of the spiders here. And we actually believe that also the diet has changed, but it's a little bit masked because of the polygon shape we observed. So the cool thing about the study is that it's done in an ecosystem research station in the middle of the Palatinate forest. There we have huge mesocosms that are 100 square meters. So we can really assess these effects in the field, more or less, in a nature-like mesocosm without the downsides of a typical field study like variation and confounding. Flooding like in Musbayata, for example. Yeah. So that was my part, and I pass over to Alex.
As you might have seen now with the Venn diagram, it changed. So I'm not into the stable isotopes research. I'm more into species interactions that could affect this biodiversity ecosystem functioning relationship and how this might be affected by different chemical stressors. And the first study I would like to highlight is right at the intersection between the ecological functions and species interactions. So it is well known that these little critters that feed on the leaf material tend to feed more on leaf material that is overgrown by fungi rather than on the pure leaf material, because the leaf material is rather hard and low in nutrients, and the fungi add like fatty acids, nutrients, etc., which is why they're also called the peanut butter on the cracker, because, I mean, we like to eat bread with jam rather than just the bread itself, because it doesn't taste very well. And it is also known that algae excrete labeled organic carbon, and this carbon can be used directly by the microbes as a carbon source, so they rather focus on this labeled organic carbon instead of the leaf material itself. And by this should also increase the growth of the fungi, thus increasing the nutritional quality of the leaves. And we did this, so we fed, we fed, so these are small organisms, the black ones are large organisms. We feed different food qualities, so pure leaf material just overgrown by fungi, and leaf material overgrown by fungi in the presence of algae to these organisms. And we saw that what we expected with increasing food quality, they were feeding a higher amount of the leaf material. And because the food quality was much higher with algae being present, they also grew faster. And this was irrespective of the size of the organisms. In a second study that goes more into this interaction or into this overlap of species interactions and ecotoxicology, in this study we only focused on the fungi growing on the leaves, and we sampled these from two distinct environments. So we had one community from a forest stream, so these haven't been exposed to fungicides or any chemicals before, and in a heavily used agricultural environment, so these organisms were pre-exposed to many chemicals before we took them into the lab. And our hypothesis was that because these organisms from the agricultural area were pre-exposed to stressors, that this community had overgone a mechanism called pollution-induced community tolerance, which means that sensitive species are gradually replaced by more tolerant species, leading to a community that is still able to maintain the functioning also under chemical stress. So we exposed these communities in the lab to different fungicides, and we actually saw that the effects were harsher for the non-exposed community compared to the pre-exposed community. So they had a much larger species turnover over the course of the experiment. And these pre-exposed organisms were still able to maintain leaf breakdown at the control level, although they were again exposed to fungicides in the lab. So this really did fit into this picture of this pollution-induced community tolerance. But this study we only conducted in Sweden. And with the third study, we wanted to go into the same direction, but cover a rather big biogeographical scale, ranging from the Fennoscandian Shield, the Fennoscandian Shield in Sweden, over the Central Plains in Denmark, all the way to the Western Highlands in Germany. So we conducted the same experiment at these 3 laboratories, again using species that were non-exposed before, took them to the laboratory, exposed them to fungicides over 3 what we called colonization cycles, which means so in the first cycle, that is here depicted in green, we used the organisms from the field to microbially colonize leaf material in absence and presence of the fungicide. And this leaf material was then used in the second cycle to colonize new field leaf material, and the same then for the third leaf cycle, for the first colonization cycle. And also here we had the hypothesis of this gradual adaptation of the community from one to the other colonization cycle, because we used the community from the previous colonization cycle. And what we found, so here we see the changes in the decomposed leaf material, and so sorry, now it becomes clearer. The solid lines are the respective control treatments, and here we have the fungicide treatments. And irrespective of the region that we were looking at, in the first cycle we always had a decrease in the function, but over the course of the experiment, the functioning kind of returned to the control level, and we also found this in the community. So we think that also here we had this gradual adaptation of the community to the environment, and with this, this like a superior or the same functional capacity.
Sorry, can you repeat what were the three numbers? 1, 2, 3.
So okay,
three different colors. Like
so the one, this is the different colonization cycles of the leaf material. For this green cycle, we took the communities from the field and colonized leaf material in the lab under control conditions and fungicide exposure. And then we used this leaf material from the first cycle to colonize new leaf material in the second cycle. And after this, we used the leaf material from the second cycle for the third cycle. So we always use the either control communities or the already exposed communities to colonize new leaf material. And so we also had a community shift from the first to the second cycle and from the second to the third cycle. So we could see this by analysis of the microbial communities. And with the last study, I would like to take you away from the lab, more into the field. This study we conducted in southern Sweden across 18 stream sites, where we wanted to distinct the effects of macroinvertebrate community, the water quality in terms of pesticide occurrence, physical-chemical parameters, nutrients, etc. Habitat quality in terms of which kind of sediment do we find in the streams? Do we have rather twigs, or do we have leaves? Do we have complete trees lying in the stream, etc.? And also, on
land use.
Sorry?
Land use.
Yeah, also land use. So, and assess these effects on the leaf litter decomposition. And we also did this with different leaf species, because leaf species differ in their nutrient content, in their toughness. So organisms tend to feed on one leaf material more than on the other leaf material. And what we found was, what was not surprising, on the one hand, that we had this effect of the leaf species identity on the leaf mass loss. This is rather known already. And we also found indirect effects of agricultural land use, or the percentage of the agricultural land use around the sampling sites on leaf mass loss. On one hand, we had a direct effect, so the higher the agricultural land use was, the lower was the abundance of sensitive macroinvertebrates, and a lot of these sensitive macroinvertebrates are known to feed on the leaf material very well, so if we don't have them anymore, we have a negative effect on the leaf decomposition. And on the other hand, the percentage of the agricultural land use affected the habitat characteristics in terms of more fine sediments being present, less leaf material being present, etc. And if these habitat characteristics are present, we also have a lower leaf decomposition, because the macroinvertebrates rather live where they have like coarse material that they can bury under, they have more leaf material, etc. And with this, we hope that you gave you a small glimpse of what we are doing back in Germany. Thank you for your attention, and we're open for questions. Yes.
No sé si tienen preguntas.
curious. That's all. I can imagine, I'm chemist.
Okay.
Sorry
Yeah, no worries.
for the mistake. I'm curious to know your appreciation if there is some difference about the exposure with the climate compared what you are seeing here with what we have done. Maybe we have longer sun radiation, the time within the day. So do you expect to see some difference with what you have done? The temperatures, the climate, the sunshine?
I would say yes, because we have longer growing periods. And for instance, fungicides are applied prophylactic. That doesn't mean we only apply them once in the growing season, but over and over again, like 10 to 12 times within the growing season. And if the growing season gets longer, there is more application. On the other hand, with climate change, we have warmer temperatures, for instance, if I talk about the northern hemisphere now, in northern region or more northern regions, meaning that, for instance, fungal pests can also travel more northwards, meaning that we have to apply more fungicides. And I would say it's the same for instance, herbicides and insecticides, because pest species move northwards because they are out of their optimal temperature range, meaning that we will apply more pesticides in the future. On the other hand, of course, there are other application techniques nowadays with, for instance, GIS systems that can identify, and then machine learning, and the algorithm can identify where we have pest plants, for instance, and then the herbicides are applied only where it has to be applied. So I think it's a trade-off between climate change making it necessary to apply more, and on the other hand, the pest management.
For example, in Valley, they but they use steroid flies to match the control the. I don't know if they're a white fly or something like this, it's a pest, and they use this non-chemical substrate to control the white flies. So yeah, it's balance between climate change effects on pest spread and the pest management techniques we are using.
And then I think it's also a matter of how new pesticide products are being designed, because exactly. So we have this one study by one of the professors in our working in the institute, and he showed that within the last 20 years, maybe 25 years, there was less insecticides being applied, but the toxic pressure was higher because they are more toxic nowadays than before. So this might also play a role in, or this might have an impact in what are we applying? Are we applying less, but maybe we have more effects because of that, because they are more potent nowadays.
Just additive. Here in Argentina, we are 10 years later with the changes of the molecules that we are using, like pesticides. So for example, imidacloprid was forbidden in 2014, the first time, and later in 2017. And we're still using it like nothing. In all the reports there is imidacloprid everywhere, here in Pampas region, Córdoba, here in Valdes.
The genetic compounds that you found are more perseverant in the environment.
Some of them, but the main problem is the toxic pressure, like you said, and the way the guys in the vineyards use it, or maybe in the, I don't know, in other croplands they use more intensively. So maybe this is the main difference. Not they are trying to convert this kind of pesticides in non-persistent ones, but the problem is the toxicity and the use.
¿Alguna pregunta más?
Okay. No hay ninguna de...
No.
So I have one for Alex and then one for...
Okay.
So you mentioned the pollution-induced tolerance.
Yeah.
And maybe you said it or I missed it, but I I didn't understand how it arises. Right?
Okay.
I can think of several mechanisms. One would be species turnover, but it could be also phenotypic plasticity or even evolution. And do you know what's going on with how you get this evolution to Stolen?
So at least in these two studies that I highlighted, we think it's mainly species replacement.
Okay.
Because we can see that the community is shifting from a more pristine state with a lot of sensitive species to a kind of adapted community with lots of species for which we know they are tolerant to these fungicides. But we didn't look into genetic differentiation or plasticity or phenotypic plasticity. So I think it's a mixture between all of these three, but in our study we could only show or could only get an indication that it was species turnover.
Okay. But some of the species are shared between the two.
Yeah, so we have species that occur both in natural or near natural communities and pre-exposed communities. But we also have some sensitive species that we didn't find in the pre-exposed community from the beginning. And these are the ones that were kind of killed in the natural community.
Okay. Thanks. For Gastón, I do it in English. So you mentioned the environmental risk, this sum of RQ, and if I understood it correctly, it should correlate with the concentration of the compounds, because you're not using information on which organisms you have in each stream or river.
No.
So should it correlate always with the concentration?
Yeah, it's a relation between the concentration we found in the environment and the concentration we know by bibliography, by the databases of other species, a lot of species that we find LC50, delta concentration 50, or maybe no effect concentrations. So it's like in the, let me show, I don't have it, but it's like a quotient between both concentration. With sometimes we use an assessment factor because of the amount of data we have, toxicity data, but this is the first year, is the first step in the risk assessment. Yeah. The second part in the paper, we make some species sensitivity distribution curves, but not with the toxicity data from the field. These data come from laboratory studies, and you use it to compare theoretically what about the field concentration of that pesticide or that mixture, but it's not about the data of toxicity data in the field.
Okay.
The potential.
Now, yeah, the potential. Now we are looking for some changes in the structure of the communities, benthos structure, and the phytoplankton. So this is the second step. I don't have enough data right now to tell you.
I mean, this is a problem with risk or with first-year risk assessment anyways. So when you want to register a new product, it has to undergo a risk assessment. And in the first year, they are testing fish, algae, and invertebrates, but only one species of each of the group, and assuming that this is the most sensitive species of the group. So, and then adding, of course, assessment factors to that for interspecies differences in sensitivity.
Most of the time, we use species from the North Hemisphere just because they perform more toxicity assays with the species with different type stories or exposure stories than in the South Hemisphere. Or, for example, we have some data from local species from Pampa region, but not from Mendoza. There are not many toxicity assays with local species by the moment. So that's
in process.
So this is a problem, but this is the first step. If you find something that is a problem, for example, this kind of risk questions, you have to move to another tier, more complex, and maybe try to make some specific essays to look into the problem.
¿Otra pregunta?
Yo tengo un par. So Gastón, it's more like directly on your presentation, but you mentioned that here the plants where they stabilize water or do not have the mechanisms to make sure those chemicals are out of the water that's used again or something like that. It's hard for me to say it in English, but are there mechanisms to do that? It's just that we are not using them here.
The problem is that all the compounds have different behavior according to, there are a lot of studies trying to degrade some specific PDCP or any compound, but they don't go for the same pathway because some of the concentration you find in the environment is product not only because of the use, but sometimes because of the process they suffer and the structure of the molecule. That is typically, for example, carbamazepine. You can find is anticonvulsant. Carbamazepine. Un anticonvulsivo, la carbamazepina, that you can find in a lot of environments. It's not the most used compound, but it's very stable for the wastewater treatment plants processes. So there are lots of studies. I'm not starting that, but the problem the focus, not the problem, but the focus here with the wastewater plants is to remove organic material and bacteria and this stuff. Here in Mendoza, you have plants that are working better than in other provinces in Argentina. I know because I'm from I know how it works. You have like a water culture here, so I could say that you are not in the worst scenario here in Argentina, of course, because they try to. We were studying in Uco Valley because here the wastewater treatment plants effluents are used in the acres, this kind of pilot fields to grow vegetables. I don't know, garlic and this kind of stuff. So most of the water is not going directly to the river or so. But yeah, there are a lot of processes to remove it, but they are expensive too. And if you don't consider that it's a problem, you are not investing this money for that.
So it's
¿Otra pregunta?
but so out of curiosity, I think Eric mentioned that fungus are the main source of these essential fatty acids, right? Oh, you did.
Okay.
Sorry. So because what I understood, I'm thinking of riparian predators, that aquatic prey are much more nutritious just because of that. And so apparently I didn't know it's because of the fungus producing those essential fatty acids or not.
Fungi produce some of the fatty acids, but these really like super essential fatty acids that are needed for reproduction, for growth, for development, etc. They are rather produced by algae.
Okay.
So algae are the best when it comes to fatty acid production. In our experiment, we couldn't find algae attached to the leaf material. That would have been the ideal case for us. So we are going more into that direction of trying to not only have the fungi on the leaf material, but also algae, and see then how the physiology of the consumers are changing because of that. And ideally, we would use, for instance, leaf-shredding insects that emerge from the water and then are being predated on by some riparian predators, just to see if their quality would change because of feeding on different quality of food sources.
Yeah, so the question was
okay.
into that direction, and I was going to ask you if for some reason pollution or whatever, fungi disappear what would you expect? So, but not necessarily fungi disappear. Also, you have algae that means we don't know if you use pesticides.
Yeah, so this would also be one of the next steps to
Okay.
kind of put chemicals in the system to see how the microbial community is changing, and with this, how the quality, for instance, in terms of the entire amount of lipids or specific fatty acids or amino acids or whatsoever you want to look at is changing.
Maybe Seth will fit to your seminar in 2 weeks.
2 weeks?
2 weeks.
Seth is the expert on fatty acids. So.
Oh, okay.
Next.
¿Alguna otra pregunta? Bueno.
Muchas gracias.
Gracias.